All right, good afternoon, everyone. And again, thank you all for coming to Hologic's afternoon symposium today. We are excited to be able to share some performance evaluation data on the upcoming Aptima CMV Quant assay. We are excitedly awaiting FDA approval. And so the data today are being presented from research use only studies, and the assay is not yet currently approved for sale in the US, as the disclaimer. So today I'm going to be sharing data from a performance evaluation study of the Aptima CMV assay for the detection and quantitation of CMV in plasma samples. And this data is being presented later this afternoon by Dr. Soya Sam, who works with Dr. Angie Caliendo and Ralph Rogers at Miriam Hospital and the School of Medicine at Brown University. And the study was done in collaboration with Dr. Colleen Kraft and Jessica Ingersoll at Emory University. All right, so I know that being in a room full of very experienced and very intelligent clinical virologists, I don't need to go into a long background and explanation of cytomegalovirus, but I wanted to just touch on a few review things. So cytomegalovirus, or CMV, is a member of the herpes virus family and is an enveloped double-stranded DNA virus. CMV is a very common virus. It's spread by close contact via saliva, urine, or other bodily fluids. And primary infection typically causes very mild symptoms, if any symptoms really at all, very similar to Epstein-Barr virus and mononucleosis, with fever, sore throat, and fatigue. And CMV, being a member of the herpes virus family, and as with all herpes viruses, establishes a lifelong latent infection within the host. And CDC and WHO estimate that by the age of 40, greater than 50% of adults have probably experienced some sort of CMV infection in their lifetime. In people with healthy immune systems, the virus remains dormant and does not typically cause any further disease. However, in people with compromised immune systems, like those that have undergone transplant, either solid organ transplant or hematopoietic stem cell transplant, these folks are at significant risk for CMV disease. In fact, rates of CMV in transplant patients are as high as 30% in solid organ transplants and can be as high as 70% in stem cell transplant. Post-transplant, patients are placed on immunosuppressive drugs that lowers the body's ability to reject the transplanted organ or cells, but on the flip side, this also weakens their immune system and their ability to fight off infections. So CMV disease can occur either by primary infection, where both the donor and the recipient have never had a prior CMV infection, or reactivation of latent infection in the recipient that has had some prior exposure to CMV, or it can be transmitted via the transplanted organ or cells from the donor and then reactivate in the recipient. And CMV disease can really affect almost every organ of the body. It can cause encephalitis, it can cause serious respiratory illness, hepatitis, gastroenteritis, and untreated infection can lead to very severe disease, transplant rejection, and even death. So to ensure that patients are able to have the best opportunity for a successful transplant and ultimately be able to go on and live long and healthy lives, we need to utilize diagnostic tools to effectively and accurately monitor for CMV infection in this patient population. And current guidelines for the management of CMV in transplant patients, as described by the Transplant Society, IDSA, and American Society of Transplantation, all recommend that nucleic acid amplification tests are used for monitoring CMV in patients post-transplant. They recommend that CMV testing by molecular methods are used to determine when preemptive therapy should be initiated in these patients, to detect for active infection and disease, as well as to monitor a patient's response to therapy. For example, when prophylaxis may need to be discontinued or whether there might be potential resistance to the current antivirals that are used. The first 90 days post-transplant tends to be the most critical window for monitoring for CMV. And viral load monitoring is typically performed at least weekly for that first 3-month window, and then usually monthly thereafter for at least a year, sometimes longer. And when patients are on antiviral therapy and being actively monitored, testing is typically done weekly there as well. Active CMV infection is indicated by either absolute viral load values or a rate of increase in viral load. And evidence suggests that trends in the viral load value may be more useful and valuable than a single viral load value that's used as a clinical decision point. And while higher viral load values and steep rises in viral load correlate with increased risk for severe disease, currently there are no validated CMV viral load thresholds for clinical applications such as CMV disease diagnosis or when to initiate preemptive therapy in transplant patients. So CMV NAAT assays have been evolving since they were first being used as lab-developed tests nearly 2 decades ago. And the variability of these early LDTs, as well as even early commercially available and FDA-approved assays, is very well documented. Praxitis et al. In Clinical Infectious Disease in 2016 published a very nice study to show that current CMV DNA result variability using the same clinical samples that were tested across multiple laboratory sites and across various assays, as well as using the newly available WHO international standard as a calibrator. And as you can see from the graph on the left, the data showed considerable differences in quantitation between the many assays that were evaluated, some even greater than 1 log difference. And perhaps of even more importance, this study looked at CMV quantitation variability across assays based on the assay target amplicon size. And a trend toward higher quantitative CMV DNA results was observed when using assays with progressively smaller amplicon sizes. And this can really be attributed to the vast majority of CMV DNA in plasma is cell-free and highly fragmented. Variability in assay performance can also be attributed to other factors. And so the best pathway really, as we're looking to move toward global harmonization and development and establishment of universal thresholds for CMV viral load, will really be accomplished by using assays where all of the steps are highly standardized and meet the following requirements. So assays really need to be calibrated and standardized to the first WHO international standard for CMV DNA. Assays should be targeting with a small amplicon size around the 100 base pair amplicon size. Automation of these assays from start to finish is imperative to be able to reduce the variability of different manual extraction methods, different manual pipetting methods, sample preparation, et cetera. And really commercially available assays that are FDA approved offer full analytical and clinical validation of the assay, ensuring excellent quality and reliability of results. So to get into the data and the findings from the study. So this study was done to evaluate and compare the analytical and clinical performance of the Aptima CMV Quant assay as compared to Abbott's real-time CMV assay, which is FDA approved for plasma. The study looked at evaluating analytical sensitivity, so limit of detection, limit of quantitation, as well as linearity, accuracy, specificity, and reproducibility using external panels. Clinical agreement and method correlation of viral load quantitation was evaluated using a set of clinical plasma samples, remnant samples that were submitted to Emory to their molecular lab for routine testing using their standard of care methods in-house. Those samples were stored frozen and then sent to Miriam Hospital where they were tested with both the Aptima CMV and the Abbott assays. All right, so just a little bit of an intro then on the different assays and the different platforms that they run on. So the Aptima CMV Quant assay is a nucleic acid amplification test. It uses Hologic's real-time transcription-mediated amplification technology, or real-time TMA. and it is run on the fully automated Panther instrument. The plasma sample can be loaded either in the primary blood tube after it's been spun down and centrifuged, or plasma can be transferred into a secondary tube and loaded onto the Panther system for testing. There is no batching. It is a random and continuous access instrument. So samples for CMV testing can also be run alongside all other Panther and Panther fusion assays. The Abbott real-time CMV assay is also a nucleic acid amplification test. It uses PCR technology, real-time PCR technology, and it runs on the semi-automated M2000 instrument system. For the Abbott, again, too, samples can be loaded either primary or secondary tube onto the M2000 sample processing instrument for automated nucleic acid extraction and 96 well plate preparation for off-board then real-time PCR. It is a batched testing system. Both assays use a similar sample volume. They both use 0.5 ml of plasma for testing. And the Aptima CMV on Panther, as far as throughput, 120 samples can be run in about 5 hours versus the real-time CMV on the M2000 is 96 sample results in 8 hours. Okay, so to evaluate analytical sensitivity, looking at limit of detection and limit of quantitation between both assays, the study used a exact diagnostics panel, and one of the panel samples were diluted in CMV negative EDTA plasma from 200 IU per ml down to 6.25 IU per ml. 5 replicates at each concentration was tested across 4 consecutive days for a total of 20 replicates. And overall, the 95% LOD for both assays was around 50 IU per ml. But with probit analysis showed that Aptima LOD was 31.62 IU per ml and the LOQ at 44.67 IU. And this correlates really nicely with what is included in the real-time CMV assay package insert. The reported LOD for the real-time assay is 32.1 IU per ml, and the reported limit of quant is 50 IU per ml. To look at linearity, the same exact diagnostics panel was used across the linear range of both assays. The panel range was from 2.6 to 6.6 log IU per ml, and the data shows excellent linearity across the panel range, as well as excellent correlation between the Aptima and the real-time CMV assays to the expected concentration values, with R-squared values greater than 0.99 for both assays. To assess accuracy, SarahCare's CMV DNA qualification panel was used. It is a 7 positive samples and 1 negative sample, and again, the concentration span the linear range of both assays. Each sample was tested in duplicate in both Aptima CMV as well as the real-time CMV assays. Looking at mean difference of quantitation then between Aptima and the expected results, as well as real-time and the expected results, and also between real-time and Aptima. And by expected results, so these were the results from the Roche CAP-CTM CMV assay, which is also an FDA-approved assay, as part of the package insert for this SarahCare qualification panel. And really, we saw very small mean differences between each the Aptima and the real-time assays as compared to expected, anywhere from 0.1 log to as high as 0.45 log. And overall, the mean differences were less than 0.3 log. Looking at then linear regression analysis for both the real-time and the Aptima CMV assay in the graph on the right, as compared to expected, both of them demonstrated excellent correlation, with the Aptima under-quantifying a little bit as compared to the real-time assay, but R-squared values for both were 0.992 and 0.985 for the Aptima assay. So looking at reproducibility, which is critically important, being able to get consistent and accurate results. with the assays, whether it's within site and across sites. We had a reproducibility panel that was developed internally at Hologic. It consisted of 7 panel members and was tested at each site in triplicate over 5 days. This table is including both the data from the Miriam Hospital site, which is Site 1, as well as our other PEO site at Tampa General, which Morse will speak about in the next presentation. And we looked at the mean log IU per ml for each of the panel members, as well as standard of deviation. And overall, the reproducibility was excellent, with standard deviation between 0.09 and 0.14 for Site 1, standard deviation between 0.08 and 0.15 for Site 2, and overall standard deviation less than 0.14 for both sites. Okay, so to move from analytical performance now to clinical performance, remnant plasma samples, a total of 389 remnant plasma samples that were obtained from CMV patients previously tested with a reference and standard of care CMV IVD assays at Emory University. Like I said before, were stored frozen, and then were sent to Miriam Hospital for testing with both the Aptima Quant CMV and the Aptima real-time assays. And this was done to limit any sample handling variables as much as possible. These samples then underwent the same freeze-thaw cycles, as well as were tested pretty much concurrently from one test to the next. Out of the 389, we have a total of 363 that were used in the agreement analysis. We excluded 26 samples. 24 of them were excluded because of instrument errors that resulted in invalids, and an additional 2 were excluded for insufficient volume. There were a total of 279 samples that were detected by both real-time and Aptima for a positive % agreement of 96.5. and a total of 71 samples that were not detected by both assays, so a negative % agreement of 96. And of the discordant samples, so the false positives and false negatives, of which there are a total of 13, the majority of these were either originally negative by the initial standard of care assay, or they were detected but not quantified, so really at very, very low viral load levels. And looking at grouping these across 3 different categories, the limit of quantitation for the real-time assay and the Aptima assay are very similar, 1.7 versus 1.72 log IU per ml. So looking at grouping them in those that are not detected, those that were detected but below the limit of quantification, as well as those that were detected and quantified, you can see that the numbers between both assays are very, very similar. And then finally, for method correlation, so of the 365, we had 262 samples that had paired quantification by both the Aptima and the real-time, and so these were used in the method agreement analysis, and results show excellent correlation as demonstrated by the Deming regression with R-squared value of 0.923, a slope of 1.068, and an intercept of negative 0.313. On average, the Aptima viral load values were slightly lower than the real-time assay, but the overall difference was only 0.09 log IU and a standard error of 0.015. And the Bland-Altman analysis also shows very good agreement in quantitation between these assays with an overall bias of 0.09. And the majority of these samples falling within a half-log agreement between both assays. So in conclusion, the Aptima CMV Quant assay has performance characteristics that are comparable, very comparable with the Abbott real-time assay for the monitoring of CMV viral loads in plasma samples. This is illustrated with a very good correlation with limit of detection and limit of quantitation, with linearity and accuracy, reproducibility within site and across sites, as well as clinical agreement and correlation of viral quantitation. And the findings of this study then demonstrate that the Aptima assay is sensitive and accurate in quantifying CMV in plasma samples on the fully automated Panther system. And I just want to thank Dr. Caliendo, Dr. Sam, and Dr. Colleen Kraft for collaborating with us on this great study and for allowing me the privilege to be able to present the data to you all here today. So thank you.